So let's get started. The echo. I'm not feeling that great, so I'll sit for some part of this, and mostly slides and some hands-on stuff. So, and I'll talk about what papers are we likely to read, and yeah, you know, the kind of minor project you will do in phases. So So just a few resources, right? So for PCA, right? Or forget PCA. Like everybody should watch this guy. His name is Grant Sanderson. How many people have heard of 3 Blue, 1 Brown? Yeah. So like I would highly recommend this playlist, Essence of Linear Algebra. Like I have not come across, like I've been taught by some great teachers, but the way this guy teaches is just like another level. So if you want to really, really understand something in depth, highly recommend this. So in the first half, we talked about dimensionality reduction and linear and nonlinear. Linear was PCA, nonlinear was TSNE and UMAP. So and I talked about orthogonality, orthogonal vectors. So all of this you will understand here. And there's something called SVD, singular value decomposition, which breaks a matrix of higher dimensions into two matrices of lower dimensions. So all of that, I mean, they're each is about like 8 to 10 to 12 minutes, and there are quite a few, 16 videos, but can't recommend it enough. Okay, so this is for linear algebra. Uh, for statistics, most of you, I think bar one, have watched the Josh Starmer. So this is run by a guy called Josh Starmer, and I think some of you have watched him before. He does this thing called BAM and double BAM and triple BAM. So that's just his way of making it more fun and musical. So for statistics, I would recommend this. Right, what is a uh, p-value and all that. So he has like tons and tons of videos, and he'll try to make it fun because, so, logistic regression. So that is for statistics. And this is the definitive guide on single cell. Uh, there's a guy in Germany who is kind of the guru of single cell. His name is Fabian Thies, right? Huh? What happened? I didn't get. Okay, okay. Sorry, I was too absorbed to get the joke. So his lab has put together this website called sc-best-practices.org. So probably way more detail than you will need in this course, but each of these tiny things we just talked for like a couple of minutes um, are covered in detail. So like QC, okay, I just saw this and remembered I forgot normalization. So we're going to briefly talk about normalization now, and highly recommend this also, right? So he is like detailed code and text for explaining why normalization is important, right? And finally, I haven't finalized, but I'll share by end of today. So most probably it'll be for the two papers that we are going to try to reproduce figure one of the two, right? I wanted to take a straw poll. So this is a paper on cancer cell lines. It's a Nature Genetics, very prestigious paper, 2020 paper. So that is one option, right? I'll let you quickly skim through the abstract. Right? So basically, they use single cell RNA-seq to profile around 200 cancer cell lines across 22 cancer types, and they uncover gene expression programs which are heterogeneous within the 22 within the 200 cell lines. Right. So we'll try to reproduce this, right? Essentially, figure 1B, right? And maybe C also, right? So that is one option. This is cancer cell lines, right? The other option is human primary T cells. So this is a 2022 science paper, right? This is the paper that I've been tentatively planning, right? Um, so they have an optimized method to perform genome-wide CRISPR activation and CRISPR interference screens on primary human T cells, right? So we will, as a class, pick one of the two, right? If you want to do the paper which is different from the what the class has picked, you are free to do so. Just that you won't like then you are kind of doing it on your own, and which is perfectly fine. If you're in like if the class picks cancer and you are interested more in T cells, you are welcome to do so. But you might have to figure out stuff on your own, because in class time will be devoted to what the class has picked, right? And I'm not, you can, you don't have to give me an answer now. I'm going to send a list with these two and maybe one or two more, right? And we'll do like a voting, right? Uh, to decide. Again, this uses single cell RNA-seq, something called CRISPR. So perturb-seq is, how many people know what is a KO, a knockout? Show of hands, right? You take a wild type cell and knock out or knock in is AI, but you knock in some gene to, right? So imagine perturb-seq. How many people have heard of perturb-seq? Anybody? Perturb-seq is basically a mixture of doing a KO followed by single cell RNA-seq, right? You want to check, but you want to perturb a single gene, right? And see what effect it causes, right? So they use perturb-seq for CRISPR activation in particular, and investigate regulators of cytokine productions. So I am personally think this might be a better choice, but I'm gonna send out a formal list and take a formal poll, right? So this was one more. So I'm gonna so now for the rest of the class we'll do two things. I'll quickly talk about scRNA normalization, and then we'll do some Python stuff which you will need. So for reproducing figure 1 of this paper, right, you will need something called as Python list comprehension. You'll need a bunch of things, but this is an important concept which I didn't cover in last class, which I think deserves to be covered in some detail. So we'll talk about normalization and uh, Python list comprehension. Okay. List comprehension, you will try out on your laptops also, right? So let's get started. gonna take an example from the video. Just give me a sec. Gonna, you've seen uh, the exact name. One to 20,000. Gonna this sample one. So, right, we did RNA-seq, right? And let's just focus on, let's for example, two samples, right? So there are two columns in our single cell RNA matrix, right? And I have only whatever 6 genes, right? A1BG, A1BG hyphen AS1, A1CF, A2M, A2M hyphen AS1, A2ML1, right? And what is important is the total number of reads for this sample across all the 20,000 genes are 635, and there is 1270. So in practice, it will not be like always a nice multiple of, this is a contrived example, but what we are trying to say is that when if you were to build a matrix or analyze, compare sample one, these are read counts, right? This with this, it's not an apples to apples comparison, right? You have 635 reads here. Here you have the exact double, 1270, right? So how do you kind of do an apples to apple comparison? You do what is normalization. So what will you do is, this is the read count for A1BG, right? You will divide this by 635 and you will divide this by 1270 to get a normalized number of reads per gene for A1BG, right? And they will be the exact same, because 30 divided by 635, if you multiply by that fraction by 2, both in numerator and denominator, you will get 60 divided by 12, right? Otherwise, you are unfairly getting the impression that this has more expression, right? So we'll not go into details, but what is happening is some samples are landing on a flow cell and some are not, right? So that should not dictate whether a gene is highly expressed or not, right? Right? So similarly, this would be 24 by 635. And this is uh, one way of doing normalization. There are more involved and complicated ways. So that is about normalization. Any questions on normalization? Okay. Now we'll. Can you guys all open your WSL prompt, like Unix or Ubuntu prompt? So I'm going to first do whiteboard and then we'll try out, right? Uh, so let's say we talked about lists quite a bit, right? So let's say you have uh, a list and let's say, okay, I'll start something even basic. I want to generate a list from 1 unto 100. Right? One option is I type L is equal to square bracket 1 comma 2 comma 200. Now, if I don't want to do that, uh, is there an so people who have done Python before, feel free to shout out the answer or volunteer an answer. Is there a way to write one line of code where I don't have to type 1, 2, 3, 4, so on up till 100 and I can generate a list automatically? Anybody? Yeah. Okay. Without, okay, for loop is one answer, but let's say if I say without writing a for loop, like literally one line of code. For loop is more than one line, right? So we'll use something called list comprehension. So what is a list comprehension? So it's a very powerful concept. It will be used in single cell RNA-seq also, because we'll have a lot of files. Not all of them we need to use for our downstream analysis. We have to, based on certain conditions, only pick a subset of them. And list comprehensions will be very critical to do that. So, so to do this, and then I'll get down to the formal definition, I'll write square bracket. Start of a list is always square bracket, right? I'll say X. We will use some for loop notation. So we'll say X for X in range 100 parenthesis. So let's try that. To make it simpler. Did everybody see that, right? So it will always be some variable which represents the list index, right? For X in something, and there might be an optional if, right? So just let me pick a more So now, right, can everybody also keep, I mean, don't just look at the screen, also try it out. Now, I have 0 through 9, right? Now, I want a list which only contains even numbers, right? So you will have to use some sort of if condition. You can scroll up and down using up and down arrow. So scroll up, right? And to this add. So just some Python syntax, right? To test evenness of a number, you need to check remainder on division by 2. If remainder on division by 2 is 0, it's an even, right? So you will say if x remainder is like remainder after integer division is percentage. So you say x percentage, what you divide by equal to 0, right? So if I do just one second. So if you have a complicated condition, you can enclose it in parentheses. Now do x. Yeah. Can you use the mic? Just press the button. No, press the button and keep it pressed. No, this one.
When I'm using the command X for X in range, it
Right.
shows there's some error and it's highlighting the parentheses as the error.
Can you double check?
Yeah. I tried adding a space, removing a space, it still shows that as an error.
Oh, after range, you have to give parentheses, right?
Yeah, I tried that. Then I thought,
There
okay,
has to be
maybe
no
there is.
parentheses, no space between range and the opening parentheses. Try that.
So this,
Yeah.
it still showed the error. Which is why I tried different activations of that.
Gonna cheat and ask Claude. So this works on Mac perfectly. Why is it not? Strange. I'm gonna cheat and ask. No, control D. You get 3.10.12. What what was the error you get? Oh, okay, my bad. I didn't spot it even. This is not the Python prompt. This is the bash prompt. You have to type Python 3 and go to the Python prompt. Triple greater than. Now type this. Yeah. Is everybody able to get even numbers? So I should be able to. Does it work now, Manasa? Oh, okay. Right. So always, I should have paid attention. This is the Python prompt, right? This is the bash prompt, right? So you were typing it on the bash prompt, right? Uh, so it was trying to interpret it as a shell command instead of a Python. So everybody was able to do this also, right? The You shouldn't give x equal to x, like give it a different name, the list, right? Like if I want everything other than 4, can anybody tell me what um, can I give here? Second. Do people know what is logical or logical and? Does everybody know that? If I want everything other than 4, I can say x not equal to 4, right? Now to give logical and, you have to do this. You have to enclose. So this outer parenthesis for the if condition, and the inner is for x not equal to 4, this condition, and x percentage 2. In single and. Everybody able to do this? So we are going to type If you want to execute a Unix command, which you type on this from the Python prompt, I am going to show you and then build on that, right? So you'll do import OS, go to your Python prompt, type import OS, right? Remember we had done os.chdir and os.get cwd, right? Last time, get current working directory, which was equivalent to typing pwd, right? So I'm going to create a directory by typing os.system mkdir. This was to create a folder or a directory. So I'll say whatever, L2, right? So it output 0. 0 means, so if a command executes successfully, it will output 0. And if I do this, so how do I check list of files? I can do something called os.listdir. So instead of typing ls, right, you see it gave me a list containing all the contents of my home directory. This is my home directory, right? I am here and I said list the contents of the current directory, right? Using os.listdir parenthesis. And the folder I had just created using mkdir, it is visible here, right? Right. Now I'm going to go one level down into that folder, right? L2 bio 450. Right? And to check whether it actually changed, I'll do get cwd. Everybody with me so far? We created a folder from the Python prompt. We we did list directory to check whether that folder got created. Then we use chdir to go into that folder. And then we verified we whether went into that folder using get cwd. Everybody with me so far? Right. Now, in this folder, I'm going to run, you remember the command to create empty files? Can anybody tell me what was the command to create empty files from the bash? Touch command. Right. So I'm going to but I'm at the Python prompt, not at the Unix prompt, right? So I'll use os.system. Whatever you type on the Unix prompt, you can pass as an argument to os.system within single or double quotes, right? So I'll say touch a.py, right? Right? 0 means it executed it correctly. I'm going to create 6 files. I'm going to create a and b.py, a and b.txt, and I'm going to create a.md, the markdown. All these are 0-byte empty files, right? Can anybody now tell me if I want to, where am I? I'll type os.getcwd. This is equivalent to typing pwd on the bash prompt, Unix prompt. So I am in this. Here I have created a.py, b.py, a.txt, b.txt, a.md, b.md, 6 files, right? Can anybody tell me what command should I use to list all these? I'll scroll up to see. Right. os.list there. Right. So let's just scrolling up. Right. Yep. Okay. Oh. Yeah. Take your time. Don't type it. I would highly encourage type it here. You're already done. So you're making a note also because you learn by writing. Check how do os dot get cwd. Right. os dot system parenthesis single quote single quote. Yeah. Yeah. So, so all the files starting with there are so dot bash rc is a file. So just ls doesn't show you hidden files which start with a dot, but list there will show all files. Now, if you uh, give it this ls, just ls will not show you system files. If you do this, right, it will list all the file types. There are a lot of files, so the first ones are not this symbol, shift of this. It's showing you this. Then if you type enter or paper, it will show you. You are following. Okay, we'll give you some time. Don't do x equal to do l equal to just shows the everybody with me so far? You have written code before, right? So everybody has the 6 files: a.py, b.py, a.txt, b.txt, a.md, b.md. Right? Now we'll use list comprehension to list, let's say, all the files that start with A. Right? This is a very simple version of what we'll do for the paper. Right? You'll have lots of directories or files, like thousands, and you want to select some subset of those files which start with a particular string. Right? So how will you do that? Right? So so what we'll do is we'll do os dot right. So let's say I call it files. I'll call my list. I'm going to make a list called files with A, right? And I'll say F if os dot list dir f, right? So I'm saying basically list files with A is F for right. This is what we executed os dot lister, right? So we are going to use it in the list comprehension. F for F in os dot lister if F dot starts with, so this is a function. So it kind of reads like English, right? I'm always focused on the F in something, right? So I'm saying run this, okay? Uh, so I'm saying run this, whatever, you will get a list from this, iterate through that list. As you iterate, whatever you call it F, and apply this condition to F that F should start with A, right? And I enclose this in square brackets, right? So if I now type files with A, similarly, you can do a files with B, right? Just switch B and call it. So technically, this thing is called an iterator, right? Your F is iterating through the output of this, which is a list, right? So, right, just to reiterate the point, right? If I do os.list there, it will list all the 6 files, right? It will output of this is a list with 6 files, right? So as you iterate through this, The iterating variable I'm calling f. Okay. Now, to make things interesting, there were two .py files, Python files, two text files .txt, and two markdown files .md, right? So just like there is a function called starts with, there's also a function called ends with, right? So can anybody volunteer? I want a function called files with py. So I want to list all the Python files in this directory. And instead of starts with, can anybody try it on their machine? What should come here? What do the Python files .py files have in common? They end in .py, right? So you will give .py. And now if I look at the variable files with py, right? Everybody able to do it? So we will kind of be using this extensively next week. Now, okay, yeah, everybody paying attention? L1 is 02468, right? Now I want to make a list which is squares of 02468. Can anybody try what should it look like? L1, help me out. How do you say square in Python? Yeah, you can do star star two, or you can do caret. This also works. Or actually, let's stick with star star two for x in what? No, no, no. The yes, right? Yeah, and let's call this L2, right? Right? Everybody following? Perfect. Right? So list comprehensions are very powerful. You can uh, add or conditions and conditions, whatever. Right? So as I said, you will have hundreds of files. Not all of them, right? It will be some complicated version of this. Can you see what's on the screen? Yeah, the file one, right? So you will say, I am interested in folders which start with raw underscore and then do some processing on them, like unzip them or something, right? Yeah. No, no, this is kind of, this I would call pre-processing. Like you have lots of data, not all of them, on some subset of them, you have need to do some processing on which you will run your ScanPy code, right? Right, right. Selecting some subset of the raw data. So in fact, the two optional samples that I had, right? This, right? If you look at this, and go to the code, right? So it's IPY is like IPython notebook. That's what it stands for. It's a Jupyter notebook, right? So I'm gonna. I was hesitant to give you this here, but basically, uh, some sort of this, right? So you have a temporary folder, right? And I have uh, just like you have zip in Windows, there's something called gunzip in Unix. So I'm gonna create files and add columns. Like this was the file. I've added a third column. I mean, he has added a third column, and for all rows, he had made it gene expression, right? Um, right. He has added like this. Opened this file to write, right? WT is write mode, and then you're doing some. So again, right? For X in files, this is again list comprehension. Not so you can use not in also, just uh, Boolean opposite of present in file, right? So it will get a little hairy, right? You have to be ready for it. We will not go into as much detail as this Sandbomics notebook, but we'll go into sufficient detail, right? And so this class is uh, kind of my way of showing you a little bit hands-on. You will do some stuff on your own also, but and the other thing you need to understand is there's this thing called pandas, which in Python is abbreviated as PD, right? So you do import pandas as PD, and then if you search for PD dot, you will find a lot of right. So a matrix or a CSV file, matrix is usually stored as a CSV file. So you use PD dot read CSV to read in a CSV, right? And yeah, and to concatenate two CSVs or add a column, you will use pd.concat. So you will plot histograms. Okay. What else did I want to cover? So for example, uh, so let's say you want to split a string, right? You can say, can anybody tell me what the output of y will be? Yes. Right. So this is y, right? And the other thing I wanted to quickly show you is just thinking how to is this function called split, right? Uh, So kind of blanking on how to use split. I'm going to take so let's say I have a sentence and I want to get the words out of it. So you can say x dot split, and you notice the 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 character separating the words is a blank space, right? So if I say x dot split, split on the blank space and store this in y, can anybody tell me what will y contain? Right. Let's call this z. Okay, let's not call it its own. It's a string method. It's not a list method. So I'll remove the parenthesis. Strings are delineated by single quotes or double quotes, right? So let's call this x, and I'll say, right? So it will be a list. You split the string, which is the whole sentence, the quick prawn fox, on uh, on a space, and whatever you get, you store it as a list. So Z is a list with splitting splitting this string, right? This string over this thing, right? And you don't necessarily have to split on empty space. You can split on anything, right? Um, I'm not sure what the output of this will be. Can anybody tell me if I just give no, what will it be? And let's call it Z1. So it diffuses. But let's say it was if I put randomly quotations, colons, and now I split on colon. What will be Z2? Anybody? So what is happening is um, it is it has created only one thing, right? It has taken the first colon, right? It has taken the stuff before the colon, and I don't know why this is a little off. Oh, I did x dot split. I should do x2 dot split. Sorry, my bad. This makes sense, right? So it splits on colon, right? Whatever is before the colon, before the first colon becomes the first element. Whatever is after the first colon, two the beginning of the second colon becomes the second element. Whatever is after the second colon to the beginning of the third colon becomes the third element, and so on. Everybody got it? Right? So split is also very powerful. Uh, and I'm not feeling too well, so I think we'll end a little bit early, maybe in 5 minutes from now. Um, so, any questions so far? Doubts? So yeah, that's good. So there is a. Uh, can anybody guess what's the antonym of split? Join, right? Very good question. So it does. So what I'll do is I'll call z2 dot join. So technically, this thing you pass to split or join is called a separator, right? So I'll do z2 and call it x3. Right? So what I'm saying is take z2 is a list, right? I'm saying take the first element and the second element of z2, combine them using a separator, what I have passed, which is colon, right? Then take the second element and the third element, combine them using the same separator, right? Uh, List. So I think so. Join only works on strings. So what we'll do is we'll have to, since z2 is a list, we'll have to do string, right? Z2 is a list. String of Z2 converts. So I'm gonna. So anytime you are unsure what data type you are dealing with, you do type of. So type of Z2 is a list. Now if I do Z3 is equal to string of Z2, right? And I do type Z3, it will be class string, right? And now let's see what. So Z2 was this, and Z3 is this, right? This is not what I wanted, but let's see if it works. Or actually, I think I made a mistake. So what we are going to do is not string. We'll do, uh, we'll use a list comprehension. So we'll do uh, x dot join, or sorry, let's do I dot join, right? For I in, can anybody tell me what should come here? What is the list? Z2, right? And just to show you, Z2 was this, and X3 is, oh, I think yeah, I'll have to. Oh, sorry, I was. So you give the separator dot join. So you have to do. Uh, and here you give I. Didn't do exactly what we wanted. Split list. So okay, let's start from the beginning. What did we do? We had. I think now we can mostly do it. X2 was this, right? And we did Z2 was. What was Z2? Z2 was this. Now we do Z3 is equal to. Okay. It we recreated it. Okay. Now is it clear? So we started with X2, which had colons in every word. We split it on colon. We got a list, which is a list of strings split on colon, right? Now, if you want to reverse, the syntax is the separator dot join and pass the argument that is passed is the list which is split, right? So separator is colon. Now I could also do, right? Could do anything, right? And call it Z4, right? Yeah, right. It is taking this list Z2 and you know insert like inserting this between first and second and third and making it a string. No, no. So it is putting, look at this. It is putting between elements of this, right? And there was a space. Do you see the space between E and Q? So that space is coming here also. It is taking this quite literally, right? Like it'll take the space as space, right? Uh, like if I were to make a small example, the quick so here there are no spaces, right, in the letters, and I so output of join is will always be a string. Z5 is a list. Z6 will be, so I'm giving it 4 underscores and saying join. What you pass to join is a list. So Z6 will be a string. Beyond? No, there's no space here. Where's the space? between first and second element, second and third, third and fourth, up till the end of the list. Right. So I think we'll end here. I'm not feeling too great. So, but I'll send out an email either today or latest by tomorrow with the list of papers and a poll. So you can vote which paper you would like to work on. And the goal is to recreate figure one. Okay.